Good morning, good afternoon, good evening, everyone. Thank you so much for joining us for this Cytobank's first webinar of the new year. My name is Liz Johannesson, and I'm the webinar lead here at Cytobank. I'd like to take a moment to introduce our senior application scientist, Qianjun Zhang, who will be leading us today. Take it away, Qianjun.
Welcome, everyone. Thank you for joining us for today's webinar. We are honored to have our guest speakers, Dr. Shahram, Kordasti, and Dr. Giovanni Povoleri to join us. So Dr. Shahram is Associate Professor and the Group Leader in Applied Cancer Immunopathology, System Cancer Immunology Lab, Comprehensive Cancer Center at King's College London. And Dr. Giovanni Povoleri is a Postdoctoral Research Associate Center for Inflammation Biology and Cancer Immunology at King's College London as well. And Dr. Shahram is our regular guest now, and you may find links to his past webinars recording about deep profiling Tregs in aplastic anemia and another talk about cytocluster R in my later slides. So for today's webinar, I will give an overview of Cytobank, introduce some of the machine learning tools that Dr. Shahram and Dr. Giovanni will present in their talk, how Cytobank can help achieve those results. We will then show the presentation of their work. They will talk about the recent Nature Immunology publication of how they use Cytobank's machine learning tools to identify novel Treg subsets and use different single-cell technologies, including RNASeq, to validate the findings. So they will join us live for the Q&A after. You may submit your questions anytime during the talk. So for those who are not familiar with Cytobank, it's a cloud-based single-cell analysis solution focusing on high-dimensional data. So before I dive into Cytobank, I'd like to set the stage for how modern technologies have shaped the world of single-cell analysis and changed the pattern of data analysis. So in the field of single-cell analysis, cytomics have been used quite often in the recent years with the development of cell analytical tools, no matter it's flow cytometry, mass cytometry, or genomic tools. We were able to now measure all features of all cells within our biological system. So here's an example showing how the myeloid cell system, before the age of high-dimensional analysis, we were only able to identify some of the major subsets. And now with the modern computational techniques, we are able to deep profiling the whole system in more details and making new discoveries. For attributes to the high-dimensional data and the advancement technology in the field, from the early 4 colors in the early 90s now, we are able to measure 10 to 20 color experiment become such a norm for many immunologists and scientists. So with the introduction of some of the newer instruments like SPADE, you can go up to 28 colors for conventional flow cytometry, and the full spectral cytometer from Cytobank overcomes the bottleneck of composition issue and able to do more than 20 colors within 2 lasers. And of course, with CyTOF, the combination of mass spectrometry and flow cytometry technology, which has really pushed the field of single-cell analysis toward high-dimensional. Theoretically, the latest heroes can measure up to 100 parameters. That's the number of channels that's available you can use. And it has been routinely used for measure up to 50 parameters simultaneously. So the whole field is going toward high dimensional and high throughput, not only the increase of the number of markers, but also the number of samples collected and the number of events collected. Now, many of the clinical trial experiments generate gigabytes of data. This really imposes huge challenge on data analysis. So I'd like to use this blind man and an elephant as an analogy that high-dimensional data is difficult to visualize. You probably are all very familiar with this proverb. It's a story about a group of blind men who have never come across an elephant before, who learn and conceptualize what the elephant is like by touching it. But each blind man can only feel a different part of the elephant body, but only one part, such as the side or the tail. They then describe the elephant based on their limited experience and their description of the elephant. So high-dimensional data is kind of like this elephant. It's an enormous amount of information. We need to combine information from all dimensions in order to be confident and make the true discovery that this is indeed an elephant. So to achieve this, compared to the traditional manual gating with low parameter data, we seek help from machine learning algorithms for high-dimensional data. So Cytobank, the cloud-based platform, is designed to meet this demand. It is designed for big data management. You can access your data from any web-enabled device. All experiments are automatically backed up on the Amazon AWS server. With this design, you can easily share and collaborate with your colleagues without the need to upload and download the data to a different device. You can share your experiment or your project with your colleagues who may work in another campus or location from your account by only one single click. So within Cytobank, we have implemented many popular machine learning algorithms like viSNE, SPADE, CyTOF, and FlowSum. You can employ the cloud compute resource to replace the local computing for those compute-intensive algorithms. And we have designed a very user-friendly interface where biologists can perform analysis without prior knowledge of R or any other coding languages. Of course, we also provide an API interface where bioinformatician or data scientists can interact with data within Cytobank to extend the application, which I will talk more in a moment. Other than the analysis platform, we provide informatic consulting services where we help clients to analyze their data, whether it's building a new analysis pipeline or any customized work. You can get in touch with us for more information in regard to the service. So in the next few slides, I will give an overview of the tools, the machine learning tools that were used in Dr. Shahram, the Nature Immunology paper. For those that are new to machine learning algorithm, viSNE is a dimension reduction algorithm that creates a two-dimension view for your high-dimensional data. So here is a viSNE map from Dr. Giovanni's paper. Basically, it plots the data on this two-dimensional plots, t-SNE1 and t-SNE2, then group the events by the expression level of the marker from all channels. And each dot here is a cell or an event. These cells close to each other have similar biological expression than the cells far apart. You can also color the dots of event by additional channel. In this case, they use CD161 to identify this CD161 positive Tregs within the T cells. So this is the viSNE map, make it easy for scientists to visually identify interesting and rare biological subsets. Of course, you will hear more about their findings later in the presentation. I will show you a animated video about how the viSNE work. So all the cells starts as one group and then scatter out to find the position on this 2D scatter based on the similarity of their expression level. So a process we call iteration. So each step is one iteration. For the cells that are similar together, they will grouped together in one subset in the unique island. For the cells that are different, there will be a different island. So this is a viSNE algorithm for to get a well-defined viSNE map. Normally, there's a few fine-tuning is required. There are 3 settings you can tweak for the viSNE run in Cytobank. One is perplexity, iteration, and theta. So here you will see, with different perplexity value, you can achieve different resolution of your viSNE map. The perplexity controls how many neighbors of any given event will have on the map. When it goes too low, you can resolve the map, and higher, sometimes not always better, because not only increase the runtime, but also not necessarily give a better results. So with Cytobank's cloud computing, you are able to set up a few different parallel runs to fine-tuning the parameters for better visualization. It also enables you to run more events, up to 2 million events on the enterprise sites. And most importantly, you will be able to build an analysis pipeline with one or more algorithm together. And you will see how that works with real case in Dr. Giovanni and Dr. Shahram's presentation in a moment. So here is another tool they use in the paper, SPADE. It's a hierarchical cluster tool that was first developed for mass cytometry data and adopted for flow cytometry as well. So what shows here is a minimum spanning tree from the figure of the immunology, Nature Immunology paper, where they run SPADE on viSNE to identify distinct T-cell clusters. In this SPADE analysis, where each node is a cluster of cells, and the size of the node reflects the relevant number of the events. And also, each bubble is a group of nodes that have similar expression level. So in this case, they cluster the T cells into 15 nodes and group into 4 distinct subsets: naive T cells, naive Tregs, memory Tregs, and memory T cells. And within the memory Treg, they were able to identify the CD161 positive Treg, which were the main target they were investigating. That in their finding, they found it's have a key role in the wound repair for the immune function. So those are the 2 main machine learning algorithms that they were used in the paper. And I would like to introduce another cluster tool that was recently introduced in Cytobank. It's called FlowSum. So FlowSum is use the self-organizing map, SUM, for clustering. SUM is a type of artificial neural network in which the nodes can be represented in a 2-dimensional grid. The SUM training process does not require any subsampling, so make it super fast. It also has a process called meta-clustering to group the cluster automatically together based on their phenotype similarity. FlowSum also has excellent visualization tools like SPADE. It uses minimum spanning tree along with other pie charts or star charts for visualization of each node. And within Cytobank, you are able to reuse an existing SUM to have persistent results between samples. And the clustering results are very comparable to SPADE, but much faster. So I would definitely recommend you to give it a try if you haven't already. One last thing I would like to touch upon is about the security of the data within Cytobank. Cytobank takes seriously about the security of user data. We use different measurements to make sure the user data is secure. But due to the time constraint, I won't go through all the details today, but you may find more information from our online security white paper, or you may write in to us for any particular question you may have. So Cytobank actually has a whole suite of data analysis solution. you can see the data. We see the data as a pipeline. The first step will be data pre-processing, which includes compensation and scale transformation, pre-gating to get a clean quality data for downstream analysis. And sample annotation is also very important, unique to Cytobank, which dictate the illustration we'll be building. And after the pre-processing, we can use either clustering or dimension reduction tools like viSNE, FlowSum, or SPADE to further visualize your data. As it is cloud-based, you can run multiple analyses simultaneously, and there's no need to take up the memory of your local computer. There are many ways you can visualize your data in Cytobank. Either you can use a heat map. When you hover over the mouse, you will see the 2D the dot plots of each subsets over here. We also have a few other tools that I would like to introduce. One is the JOP. JOP is a data conversion tool that converts any non-cytometry data into a FCS format. We can take single-cell RNA-seq data, imaging data, or any numeric spreadsheet data. So it will once it's converted to FCS file, you are able to analyze them in Cytobank for any of the tools that's available here. Another important feature we have is the Cytobank API. People can, with coding skills, can extend the functionality with this tool to intact the data stored in Cytobank. We have a few webinars that's available to use the Cytobank API in the slide. Actually, it's over here. So you will be able to, on our Cytobank YouTube channel, you'll be able to find this webinar recording about to intact the Cytobank API with the CytoCluster R, which was featured by Dr. Shahram as well. So for most of the people, we're using a normal workflow where you access the data or analysis with a web browser. But for many other people, that you can actually do a lot more with your data, where you can interact the information between the electronic lab notebook or LIMS system or any other central repository place where you can interact with the data between Cytobank and the other storage, data storage place. Or you can build a customized pipelines. In this case, you can build the CytoCluster R algorithm, which is not available in Cytobank, but you are able to interact the data within Cytobank with the API, and then after you're done with, you can feed the results back to Cytobank. So there's a lot more you can do with Cytobank with this API. We have very extensive support articles online. You may check it out to how to use this API function. And one more time, here's a summary of the data analysis pipeline within Cytobank. Once you have your hypothesis and experiment setup design after data acquisition, the first thing will be data quality control and clean up to get single live quality data. Then the data will be compensated, transformed before run machine learning algorithms. You can start to use viSNE as an exploratory data analysis tool to visualize the data on a viSNE map. Then you may use SPADE or FlowSum or even Citrus to cluster and identify significant subsets. After that, the clusters may be overlaid back to the viSNE map for further visualization and analysis. So it's a whole pipeline that you can build with the Cytobank analysis tools. That's the brief introduction to Cytobank. If you have any question, I'll be happy to take it during the QA session. But without overdue, I would like to turn the screen for Dr. Shahram and Dr. Giovanni's presentation.
All right. Good morning or good afternoon, depends which side of the pond you are. I'm Shahram Kordasti from King's College London. Today, with my colleague Giovanni, we are going to talk about the subject of Tregs in human. Perhaps you've seen our previous webinars regarding different aspects, and today we're going to review what we discussed before very briefly, and then after that the recent publication by Giovanni, and which the lead author was our collaborator and my colleague Dr. Ben Afzali from NIH, and we've been working together on this very exciting project and identified a novel subset of regulatory T cells. So when we talk about Tregs, obviously always the issue we had with regulatory T cells in human is CD4 T cell plasticity, and CD4s are quite flexible, if you like, or very easily could switch their phenotype, depends on environment and transcription factors, and they can switch from pro-inflammatory to suppressive phenotype and function, and that is an issue how we define them. Also, the way we define Tregs is quite challenging. Perhaps you may know or not, but I'm a hematologist and my background is bone marrow failure syndromes. And one of the example of bone marrow failure syndrome, and one of the most common, one of the common causes of bone marrow failure is aplastic anemia, which is a rare disease. And we have shown that regulatory T cells are reduced in number and function in these patients, and lack of function and number of Tregs can be a predictive biomarker for response to therapy in these patients. Nevertheless, what we have noticed is how we define Tregs. And if you look at this figure, I asked 3 experts in the field of regulatory T cells and asked them to actually gate Tregs for me based on 2 very well-defined markers for regulatory T cells, which is IL-7 receptor or CD127, and FOXP3, which is a transcription factor. And generally we say FOXP3 positive 127 low expression is a marker for regulatory T cells. But when I asked these 3 experts to gate regulatory T cells for me, as you can see here, we have 3 different kind of gates, which not necessarily wrong, but it's a very kind of personal decision or personal choice of what we define as FOXP3 high or 127 low. As long as you do an experiment within your own lab and you compare your results within your lab, perhaps it's not an issue as such. But if you want to compare your result between centers or do multi-center study, that's when you have the issue and you cannot compare your results. And if you go to Treg subpopulation, that is even more difficult, because you may heard that regulatory T cells based on expression of CD45RA and FOXP3 can be divided to 3 main population, which they call it Treg 1, 2, or 3. And of course, the question is, where do we really put these gates? And if you change these gates, you can have different results. And again, it's very difficult to compare results between centers. So to overcome this issue, a few years ago, we started using mass cytometry or CyTOF to see whether we can identify Tregs and define them a bit more consistent and a bit easier, if you like, and more comparable between centers. And we use this pipeline of dimension reduction by stochastic neighboring embedding using a clustering and hierarchical clustering to identify the markers, the population of cells that we are interested in. And this is published by Kirsten from Jonathan Irish lab in Vanderbilt University first, and then we adopt this pipeline in collaboration with Jonathan. And basically, you do generate your data, do quality control, dimension reduction and visualization, and then analyze data. And based on that, this is an example that published by Jonathan, and you can identify different subpopulation of myeloid malignancy, a type of myeloid malignancy, which is acute myeloid leukemia. You can see the malignant cells, immune cells, and what do they express, etc. And a very talented bioinformatician in my group, Thanos, developed a package, CytoCluster, that you can connect to Cytobank, download the data, and do the hierarchical clustering using this tool, which is freely available. And there is a webinar which is available through Cytobank as well, that Thanos explained how this works and how we can use this tool for hierarchical clustering. Anyway, these two population of Tregs that we found, which are CD25, FOXP3 high and 1 to 7 low, we have noticed in patients with aplastic anemia, which is again is my specialty clinically and the focus of the research I've been doing in the past few years. We have identified two populations of regulatory T cells. We call them Treg A or B. Some people call them naive or memory Tregs. There are some issue if you call them naive or memory, but it's up to you what you call them. But Treg A or B, they are functionally different, and the number of these cells are predictive for response to immunosuppression in some of the autoimmune diseases. And interestingly, they can be identified by a handful of markers. You don't need a very big panel to identify them. And the other aspect is when you use this pipeline for identifying these two subpopulations of Tregs, you can see that population 3, what we call as population 3 Tregs, they are a combination of non-Tregs, and the rest of them very much similar to population 2. Seems like this Treg population 3 are a combination of population 2 and non-Tregs, which are here shown by red. Therefore, more or less, you can have two main population of regulatory T cells. But when we continue in a recent work that now Giovanni is going to discuss further detail, we have noticed that these regulatory T cells within population B, or memory Tregs, if you like, they express CD161, and CD161 really is a receptor for NK cells, and basically we don't expect a regulatory T cell to express them, but they do express CD161. And this population, it was the question of what are they, how different they are from CD4 T cells that expressing CD161 and they express IL-17. Do we have a contamination of effector cells or T helper 17 cells, or actually these are a different population of cells? And that was the research question that in past couple of years Giovanni and Ben been working on, and Giovanni is now going to talk in more details. Over to you, Giovanni.
So thanks, Shahram, for the kind introduction and doing most of the heavy lifting, discussing how CyTOF analysis can be used to try and identify novel populations of T cells. So as Shahram introduced, our main interest was to determine whether this population of Treg expressing a molecule, which is CD161, has been linked to the production of the capacity of the T cells to produce IL-17, and whether this indicated that these cells were sort of contaminating of an effector population producing IL-17, or whether this was a specific and distinct population of memory Tregs. And indeed, based on the profile of the t-SNE that we just shown and the SPADE analysis indicated here, what we actually found was that we can characterize a population of CD161 positive Tregs within the memory population of Tregs. And indeed, they do express a specific profile that can be identified by mass cytometry. What was, however, an important question was whether these population could be composed by multiple subpopulations within the subpopulation, and to determine whether this population is particularly heterogeneous, so we performed single-cell RNA-seq. And however, what we found was that we could find a specific cluster of CD161 positive Tregs, which cluster closely to CD161 negative Tregs, but separated from the different populations of conventional T cells. Moreover, when we looked at the expression of the specific markers that identify Tregs, again, we can find a specific pattern of expression of multiple markers, suggesting that a population appears to be specific within the memory Treg population and is not the presence of contaminating effector T cells. However, to just try and determine whether there was a case of overlapping within CD161 expressing Tregs and conventional T cells or T effectors, we perform TCR sequencing. And here, as you can see here, all the population we analyze, so we always focus within naive Tregs, which have been defined before as population 1 of Tregs, memory Tregs, which compose the majority of population 3 as previously described, and then your CD161 positive Tregs, as well as CD161 positive conventional T cells and the totality of the conventional T cells. As you can see here, you can see that the population is mainly polyclonal, so there is not an expansion of a single cell that takes the majority of the population of CD161 positive Tregs. And when we look at the different sequences, almost 89% of the TCR sequences are unique, and they are not shared with other populations. And indeed, when we calculated the Morishita or similarity index, which compares the similarity of the different TCR repertoires, very nicely you can see, based on the TCR repertoire, CD161 positive Tregs associate quite closely with the repertoire of memory Tregs, but separated from conventional T cells, suggesting further that there is no contamination of an effector population expressing CD161, but this is a clear and distinct population of human Tregs. However, the question that we had was whether these cells are bona fide Tregs, and so to determine this, we use sort of the most common analysis that have been used to determine whether a population of cells is a Treg or not. So as you can see here, CD161 positive Tregs express FOXP3, albeit to slightly lower levels than naive memory Tregs. However, when we looked at an in vitro suppression assay, you can see that these cells are very highly suppressive, even at lower dilutions compared to the other population of naive memory Tregs. Furthermore, we did a in vivo assay to determine whether these cells were able to suppress a severe xeno-GVHD model, and indeed, when we injected expanded memory Tregs, or CD161 positive Tregs, both of these populations were able to increase the survival in this xeno-GVHD model. However, what we were interested in determining is was exactly what makes these cells unique. And one of the main aspects that really captures our interest was the fact that when you stimulate these cells are able to produce a wide array of cytokines, not only sort of suppressor molecules like IL-10, but also pro-inflammatory cytokines like IL-17, IL-22. And indeed, this wasn't limited to analysis of gene expression, but also at protein level, you can see that they produce highly significant, higher amounts of protein, both as sort of suppressor like IL-10 and IL-4, but also pro-inflammatory as IL-17 and IL-22. To determine, therefore, how this is possible, we perform ATAC sequencing, which allows to sequence the open chromatin within the cell. So we sorted the three populations of interest, so Tregs, so naive memory CD161 positive Tregs from 3 donors, and then we looked at what were the specific peaks indicating specific regulatory regions within the Tregs that make them what they are. And then we perform motif analysis to try and identify a circuitry or transcription factor that might be important for the program of these cells. And we found, interesting enough, that apart from the high expression of ROR-C, which is obviously upstream of the expression of CD161, we found there was an enrichment for the motifs for BAC2 within the CD161 positive Tregs. However, what was particularly interesting is that when we look at the expression of this transcription factor, we saw a significant increase in the expression of ROR-C in 161 positive Tregs, but significant decrease in the expression of BAC2, which makes sense considering the role of BAC2 as sort of like a regulator of the pro-inflammatory program of the cells, and it's this been described that higher expression of BAC2, especially in Tregs, sort of gives them this regulatory phenotype by blocking the effector differentiation of the cells. What we found that was particularly interesting is that when we looked for patients that presented a mutation for the BAC2, patients that presented mutation for the BAC2 presented an enrichment of CD161 positive Tregs, sort of suggesting once more that in the absence of BAC2, you might find an enhancement in the an increased percentage of these cells. We then determined to try what this program, sort of what this selected transcription factor might lead these cells to do. So what's the function of these cells? And when we look at overlapping of the different genes controlled by this transcription factor that were enriched in CD161 positive Tregs, we found that the majority of these are involved in response to wounding and wound healing. Which makes sense considering that the cytokines that we discussed before have been shown to have a role in wound healing, particularly in the gut. And I'm discussing about IL-17, IL-22 in particular. So what we did initially was trying to determine where these cells might be important for wound healing in the gut. And the first thing we did was to determine whether these cells are enriched in biopsies taken from Crohn's disease patients. And then indeed what we found was not only they were highly enriched in the colon of Crohn's disease patients, but also in healthy control they are quite enriched, suggesting that these cells might have a particular role in the maintenance of gut homeostasis. So to try and determine therefore whether this effect had a role in wound healing, what we did was to stimulate the cells and then use a supernatant in an in vitro wound healing assay to determine whether the supernatant from these cells was able to enhance the wound healing within the gut. And indeed, what we found was that when we expose a cell line from the gut to the supernatant of activated CD161 positive Tregs, we have a significant enhancement in the wound healing rate. So the wound closes significantly faster compared to the population of memory Tregs. And interestingly enough, to try and determine what was the effect and what exactly were the mediators responsible for this wound closure, we found that IL-17 and the combination of IL-17 and IL-22, which I indicated before have been described to have a role in wound healing in the gut. When these two cytokines were blocked, we could sort of slow down the wound healing enhanced potential of the supernatant containing cytokines from these memory Tregs. So to sum up what we have shown in this paper, by using an integration of multiple techniques to try and validate whether this is a population which is distinct from a population of Tregs, and we were successful in showing that this is a population of distinct memory Tregs controlled by a series of transcription factors which leads to the generation of multiple cytokines upon activation, which help wound healing within the gut. So to conclude, so sort of what is the point of this discussion is that by initially looking in sort of an unbiased manner to try and identify a single population of Tregs by using multiple markers that have been indicated as important for the clustering and allowing the discrimination of different populations of Tregs, we were indeed able to identify a distinct population of human Tregs. This finding by CyTOF was then validated by using a series of other techniques, including single-cell RNA-seq, and by applying a series of different techniques, we were able to answer different questions about what makes these cells unique and what is their function. So by focusing on protein analysis as well as genetic analysis, as well as using functional assays to determine exactly what makes this cell tick and what makes these cells unique. So this is a good example to how integrating multiple techniques allows you to give like a proper answer and try to identify the role of a specific kind of T cells in the immune system related, for instance, to a disease. Lastly, I would like to thank all the people that were involved in the study. So my supervisor, Ben, and Giovanna Lombardi, obviously our collaborator, Shahram, that very kindly introduced me to this webinar, all the funding bodies and the people that were involved in the study. And I'm happy to answer any questions.
Okay, thank you, Giovanni, and we're both happy to take questions.
Yeah, thanks for the great presentation. We now move on to the live Q&A. For those that are not familiar with Cytobank or haven't used it before, you're welcome to sign up for the free 30-day trial on our website. You can also take advantage of our training and consulting service, including the educational resources that's available on our Cytobank YouTube channel. The two of the webinars that were recently mentioned in the presentation. One is the deep profiling of the Tregs by Dr. Shahram, and also the Cytocluster R with Cytobank API. So welcome to give it a try. If you have any questions, feel free to write in to our support portal. Now I'm starting to taking questions now. So we have one question from is that what is the correlation between CyTOF protein expression and mRNA in Treg? Dr. Giovanni, would you like to take that one?
So yes, thank you for the question. So it hasn't been shown a direct correlation between the levels of messenger RNA and protein per se. So if the question is more related whether we have validated the expression of the proteins of the markers used for CyTOF within the CyTOF panels and the messenger that we done on the cells, then yes, there was a correlation between the markers that you see in a protein level by CyTOF and messenger level within the our arrays.
Great. Thanks. We have a second question coming here. Says, how did you acquire the CyTOF data? Was all the data collected in one day or over multiple days? How do you account for differences in day-to-day variation when running t-SNE on Cytobank?
The data, this is Shahram Kordasti, the data have been collected on different days, but what we usually do is, almost always we do, is that we run a known samples with our runs, same sample all the time, and we have it as a control to make sure that we can compare to that known sample, if it's necessary, normalize or check the quality of the data to minimize the batch effect. And of course, the antibodies and all the reagents are the same that we always use. So for each study, we keep the same batch of antibodies and reagents. That's what we do.
Yeah, thanks, Dr. Shahram. Yeah, batch effect is important for before the t-SNE run, so make sure your data is well quality controlled, as Dr. Shahram just mentioned, for any downstream analysis. The other question I got here, it says in Cytobank, do you recommend if different clustering and cell type ID method to give different results? Here, I'll be happy to take this one. Different cluster methods, I'm not sure if you're referring to the SPADE versus FlowSum. And in general, you know, SPADE and FlowSum can have very comparable results. There are a few literatures available to do comparison between, you know, FlowSum and SPADE. There are tiny slight difference between rare events detection. You may find we just recently had a webinar with Sophie Van Gosten, the original developer of FlowSum algorithm. You may take a look at that webinar, and if you have additional questions, feel free to write in for more discussion. We have more questions coming now. This is from David. It says, what do you think is the role of the inflammatory cytokines produced by CD161 positive Tregs? Do you think they can cause inflammation in the intestinal mucus?
So that is a very good question. So taken separately, obviously the pro-inflammatory cytokines that I indicated, like IL-17 and NR22, for instance, they can indeed cause inflammation within the intestinal mucosa. However, the combination of cytokines is particularly specific. So, for instance, if we look at the amount of cytokines that are produced, these CD161 positive Tregs produce as much IL-10, if not more, than the amount of IL-17. And we can say the same for the amount of IL-4, for instance. So what we've shown so far is that this cocktail of cytokines, so the combination of both pro-inflammatory and anti-inflammatory mediators, are what sort of leads to a wound healing effect more than an excessive inflammation. So yes, the inflammatory cytokines that are produced by the cells alone can increase inflammation within the gut, but the combination with anti-inflammatory cytokines at very high levels can then instead lead to a wound healing, specifically in the gut, considering that both IL-17 and IL-22 have been described in the gut to have a role in the wound healing. So hopefully that answers the question.
Great, thank you, Dr. Giovanni. We have one more question coming. Said, did you perhaps look at CD161 expression in NK cells?
Yes, so we have tested CD161 expression in NK cells more as sort of as a positive control as to test the expression of CD161. We have not, if I imagine that the question is within
the gut biopsies, for instance, no, we haven't looked at NKs within the gut biopsies.
I see.
But NK cells express very high levels of CD161 is one of the cells that have been initially described as expressing CD161, so for sure they will. But we haven't looked within the context of the gut.
I see. One other question related to that is, have you looked at the CD161 Tregs in other places other than the gut? And what are the clinical application of the findings that not related and not limited to gut?
That is the ongoing projects, and a few different diseases. That's what we are doing at the moment.
And one was published initially by Ben Afzali, showing that in the joints of rheumatoid arthritis patients, you can find CD161 positive Tregs. So yes, they have been described in other places.
I see. Yeah, one more question from Anna of Boston University. She asked that, is the mass cytometry data set discussed here published?
So the data that I have presented is published and is present in the Nature Immunology paper that has been reported during the presentation. The data sets per se, I don't think they have been deposited into like a online repository.
I see. So one, just I would like to ask additional follow-up with that. Do you have a plan to make the data set public?
Not in the close future, but it's something that we are considering, yeah.
Sounds good. Thank you. Here, one more question from Courtney Jackson, that in the suppression assays, are the Tregs pre-activated?
So this is a very good question. So the answer is no. So the assays, the suppression assays that have been shown here were from freshly isolated Tregs. That said, we've also tested the suppressive ability after activation, and obviously, after activation, the cells are even more suppressive.
Got you. So another question says, thanks for the presentation. I'm very curious about your RNA-seq data. Look, I'm curious to know, how did you perform the experiment, and especially the data analysis and tools you used for the RNA-seq data?
For that, I would recommend to look on the paper, because obviously the description of the experimental design would be long to do, but there is a section specifically in Materials and Method describing exactly how the data was analyzed, and actually the programs that we used, so the different algorithms and packages that have been used. So that is present in the Materials and Methods of the paper.
Great, thanks for the information. Let's see if they have additional question coming. I guess that's all for all the questions I got so far. So thanks so much, Dr. Shahram and Dr. Giovanni, for this great presentation and great findings using different single-cell techniques. I think it will be very useful for other scientists to try it out with different techniques to validate the novel findings. So thank you very much. And if you have any question later on, feel free to write in. And also, you have any question regarding to Cytobank, feel free to write in as well. So thanks so much for joining the webinar today. And,
thank you for inviting us to present.
Thank you very much.
And attending the webinar.
Thank you. It's our honor to have you. And actually, for follow-up, we will have the live webinar link sent to all the registrants, and feel free to follow up that way. Okay, thank you, everyone.